Alright everybody, this is going to be our next video on mitosis and specifically looking at eukaryotic chromosomes and differentiating the vocabulary. Let's get started on this next video. So if we're looking at the vocabulary, this is where students commonly get confused. We get the definition of a chromosome, and a chromosome is a genetic information. It's all the genetic material, the DNA bundled and packaged. Now we also get the vocabulary chromatin. And chromatin is the DNA wound around the histones. And then lastly, students are going to get this definition for what histones are. And histones are just proteins in which DNA is wound around. So really we have chromosomes, chromatin, and histones. We're going to look at an image here to try to better understand this. But what we're really looking at is hierarchy of DNA. And we're looking at either super compacted DNA or really loosely wound DNA. We're going to talk about how we can differentiate between this and how we can distinguish this and where it occurs. But the goal here is to just kind of understand the hierarchy of this DNA. Let's go take a look at this next diagram to better understand this. So if we're looking at this, what we see are differences in the DNA. So what we have here is our DNA double helix. But if we stretch out that DNA double helix, it's going to be much too long to get into the nucleus. So what our bodies have done and what our cells have done are wrapped them around these little histones here. And nucleosomes, we're referring to them as multiple histones that kind of congregate together in eights. Now these nucleosomes or these histones are going to look like this. They're all going to wind together, and then we're going to get this chromatin. And chromatin is just a single strand of these histones with the DNA wrapped around it. And then when we go through mitosis, this is when we get our chromosome. And the goal of getting this chromosome here is to ensure that everything is packaged as small as possible, so that way when we go through mitosis, we can separate and get the chromosomes that we need for both cells. So if we're thinking about hierarchy here, one, we have our DNA double helix. And then we're going to wrap that DNA double helix around proteins, which we call histones. Now we're going to take those histones, and then we're going to get to our third level here, and we're going to wind them up, and we're going to form chromatin, and we're going to form nucleosomes, which are a bunch of histones put together. Now for here in this image, what we get is further condensation of that chromatin. We can have it loosely wound so that way we can get genes expressed more easily, or tightly wound so we can get genes expressed a little bit harder. And then we finally get to our chromosome. And our chromosome only exists in this X form when we're going through mitosis, because it allows for us to go through and highly compact the chromosome to limit the amount of mutations that we have and to ensure that the cell gets both sets of chromosomes amongst the two cells.